Hello students, welcome to my talk today. So overall today I'll be giving you a bit of information on how prey deal with antipredator defenses in regard to predators and how these defenses can maybe be used to determine whether an adaptive response to stress exists within the dusky-footed woodrat. Predator-prey relationships really represent one of the most essential and important impacting factors in an ecosystem. Most often when these relationships are studied and thought of, it's in regard to the direct effects of predators consuming prey, altering their densities, and ultimately how that affects population dynamics, so how populations change size and interact with one another. However, this really represents only part of the bigger picture of what's occurring. Another major impacting factor are all the indirect effects, also known as fear effects. And these are all the effects that predators have on prey outside of direct consumption. And they're really important because the fear of predators will induce these costly anti-predator defenses. These anti-predator defenses are any way that an organism protects itself from potential predation. So this can be morphological, like this porcupine we're seeing up in the picture, and the spines that it uses to protect against the jaguar. This can be behavioral defense, so it could be increased vigilance, like we're seeing with the meerkat, where it's on the lookout for potential predators while the rest of its group forages for food. It can even be as simple as a squirrel hiding in a tree and not foraging for food, but rather avoiding predators altogether. Lastly, this can be also physiological, so the effects of fear on its system, and it's most common with the flight or fight response, where their hormones and chemicals get elevated within their system, and that allows for the temporary potential benefit to survival and escape. And so my research really focuses on solely the behavioral and physiological aspects of these defenses. We see antipredator behaviors in a number of different ways, but I'm going to tell you about some of the most common. The first is changes in time spent foraging. So they might reduce the amount of time they spent foraging to avoid predators, or forage in a new area altogether to avoid contact. Second is through habitat selection. So they might choose areas that they know are less risky for encounter, or areas that decrease vulnerability so that they can actually maybe be in an open meadow and see a predator coming from far away. We also see things like increased vigilance, so increased lookout for predators around them, or increases or changes in the group size. So a predator is less likely to come after a prey or a group that it might hurt itself in doing. However, these behaviors can come with a number of trade-offs, especially physiologically. Reduced time spent foraging can lead to potential starvation, while foraging in a new area can actually alter tissue composition, whether it's within the muscle tissue or the hair of an organism. Overall, the increased energetic costs of vigilance as well as group behavior can lead to declines in survival and ultimately impact the fecundity of an organism, so how many offspring are being produced. Predator-induced stress can come from two main sources. You have a short-term stressor, which is when prey typically first encounter a predator or a predator cue. This activates their flight or fight response and causes a massive increase in stress hormones. And this is really beneficial temporarily because it allows for the increased likelihood that they'll escape an encounter and live another day. However, if we're seeing things like repetitive short-term exposure or long-term continuous exposure, these elevated stress hormone levels remain within their system for a long period of time. And why this is beneficial if they ever encounter a predator, these continuous elevated levels can have a number of physiological consequences. We can see things like reduced growth rates, slower metabolism, as well as suppressed immune response. This can lead to vulnerability to both diseases and predators in their future, overall potentially reducing survival as well as offspring output. And so I look at this both behaviorally and physiologically, within the dusky-footed woodrat, Neotoma fuscipes. And the dusky-footed woodrat's really cool because it's a California native species living in dense chaparral habitat. It is experiencing short generation times where it really only lives about 1 to 3 years, and it's highly abundant here in Southern California. It builds these very large conspicuous stick nests known as middens, where typically 1 to 3 individuals will live. And it's a nocturnal generalist herbivore, so it goes out foraging for food at night, and it'll consume everything from seeds to flowers to woody material, and is known to be the occasional nest predator. Ultimately, it has a number of native predators. Basically, anything that eats meat will eat it. However, its primary source of mortality comes from the bobcat. My research examines exactly how N. fuscipes copes with both short and long-term perceived predation risk. And I do this by experimentally elevating predator odor in the form of bobcat urine for both wild and captive individuals. I predict that during the course of my experiment, short-term exposure to perceived risk will cause declines in foraging rates. We'll see a temporary increase in stress hormone levels due to fear, and that there will be no impacts on body condition because it is a short-term stressor. Under long-term exposure to perceived risk, I'd expect further declines in foraging rates, a continuous increase in stress hormone levels that's dramatically larger, and a decline in body condition due to, say, potential starvation from less food uptake, as well as the impacts increased stress hormones have on their physiology and body. So how do I get at this? Well, I had my study sites out in Southern California. You can see by the dot in blue, that's where we are in Northridge. The dots, or the stars in yellow, indicate my field sites, and the star in red indicates a site where I took captive individuals. So starting with the stars that are yellow, these are my 2 main field sites. Each field site was divided into 2 populations of a control population and treatment population. They were spatially isolated from one another, and no individuals moved between control and treatment populations during the course of the experiment. The red star indicates UCLA Stunt Ranch, where I took approximately 10 individuals and brought them into the lab here at CSUN for controlled stress tests. And this is really important to do sometimes in a controlled environment because it really allows us to see the fine-scale changes that occur within hormones, specifically in fecal samples, which are a really nice non-invasive way of testing for this. So once they were brought in, they were allowed 8 days to acclimate to their new surroundings. So there's stress of travel and trapping. We need to let them return to some kind of baseline level. After the 8 days occurred, I exposed them to bobcat urine as their environmental stressor, and then I collected fecal samples every 2 hours for 12 hours, and then every 12 hours for 120 hours. So now we have a really fine-scaled look at changes in stress hormones. Within the field, I exposed them to risk to see how they'll react out in nature. First, I established baseline levels. I trapped individuals, took into account sex, mass, and skull width. Mass and skull width I was able to use to create a body condition index, so if they were in better or poorer average condition. And then a fecal sample was collected from each individual to measure stress hormones. Foraging was measured using giving up densities taken for 3 consecutive nights. So what are giving up densities? Well, giving up densities measures an organism's quitting harvesting rate, which is how much of a known food source they're willing to leave behind given the amount of perceived risk they're experiencing within their environment. So basically, if they're experiencing high risk, they're not going to stay in one spot for a period of time, so they're more likely to leave greater amounts of food behind. This would be indicating a higher giving up density. That's in contrast to if they're experiencing low risk, they might eat more food. They feel relatively safe, and that'll be a lower giving up density. And I did this by setting out seed trays full of approximately 10 grams of sunflower seeds in 2 liters of sand mixed together. These were set in dense chaparral at dusk and then collected for the remaining seed at dawn. Next, I induced a treatment of bobcat urine by placing a dispenser full of it at each midden. Middens within the control patches were given a dispenser full of water. Following this, I measured the impacts of short-term risk, so how they immediately respond to that bobcat urine. And I did this by measuring, once again, giving up densities for 3 consecutive nights for foraging, and then trapping for changes in body condition and initial stress response. After 90 days of continuous exposure, I measured those same variables once more for the effects of long-term perceived risk. Once I collected all those samples, I needed to quantify stress. And how I did this was by extracting stress hormones from those fecal samples and analyzing it using a process called enzyme-linked immunosorbent assay. And this really allows us to quantify the amount that's within each sample. Ultimately, what was found? Well, let's start with foraging. I'm going to orient you to these graphs, and they're set up very similarly to one another. So on the x-axis, we have foraging or giving up density under baseline levels, after short-term stress, and after long-term stress. On the y-axis, I have giving up density in grams of seed left behind. Green is representative of treatment, and orange is control. And so what I really want you to focus on is the change in the control groups and the change within the treatment. We see that there was no significant difference in foraging under the control group. However, under treatment group, we see that there is a short-term response under that acute stress. What's really interesting to take into account is that giving up density actually decreased, which means that they were taking up more food during that period of short-term stress, which is quite unusual given that prey normally reduce foraging during increased stress, and I'll talk about why this might be later on during the discussion. Next, we had changes in condition. So our x-axis remains the same. On the y-axis, we've got body condition index. Positive values indicate better than average condition during the course of the experiment, and negative values poorer than average condition. And what's really interesting to see was that there was no significant negative effect of stress on body condition, which we expected for short-term stress. However, long-term we see a positive increase. And so it's really important to think about how 90 days have passed. And during 90 days, we actually see some seasonal changes. So it's possible that with a positive increase in condition is due to maybe better weather, increased food resource availability, and potentially a couple other things. How stressed did they get during the course of the experiment? Well, our x-axis remains the same. On the y-axis, we have stress hormones in nanograms per gram. And what I found was that there was no long-term stress response due to treatment. If you remember back when I was talking about the captive individuals, we really can't examine those fine-scale changes that initial stress causes within the field. Because field samples are taken up within about a day or 12 hours of them being captured, we can't assess those acute or short-term changes. So that's when we look at the results of the lab test of the short-term exposure to stress. And what I found was that there was a significant effect of bobcat urine approximately 4 hours after initial exposure. So we see almost an immediate response appearing within those fecal samples before it drops down and returns to baseline levels for the next 120 hours. Ultimately, what does this all mean? Let's revisit these hypotheses. So under short-term exposure to perceived risk, we found that there was no decrease in foraging rates. There was actually an increase. And this is really interesting, and it's likely due to the location of that perceived stressor, that bobcat urine, at their middens. Suddenly their midden, their nest, their home, was an area of increased perceived risk. In many cases, animals will actually spend more time away from and avoid areas of increased risk, even if it's their own nest or den. So likely they spent more time away from their middens where the seed trays were rather than close nearby. And this is a cool response that you especially see if they ever have offspring, because you don't want to lead a predator back home. We did see a temporary increase in stress hormone levels after short-term exposure, and there was no impact on body condition. When it came to long-term exposure to perceived risk, there were no declines in foraging rates, no dramatic increase in stress hormone levels due to the treatment, and no declines in body condition. So why is it that I don't see a long-term response to risk? And this is possibly due to habituation. So habituation can really be defined as a learned response to a constant stressor within an individual's environment. Habituation can be very beneficial in some cases, while it can be very harmful and maladaptive in others. In the case of constant risk, where environmental stressors and cues are continuous and predictable, it's adaptive to respond with habituation. You don't really need to respond to something that's always going to be within your environment. However, if we see things like cyclic predation risk, where there are intense periods of perceived risk within your environment, followed by no risk, followed by sudden risk, followed by no risk again, it becomes unpredictable. If you habituate to an unpredictable cue, that's more likely to be maladaptive and lead to death. You don't want to stop responding to something that may actually kill you. So do wood rats have a long-term stress response? Well, my research indicates that no, they didn't exhibit a long-term response to perceived risk. Behavior and stress responded under short-term perceived risk and then appear to have habituated under long-term risk. Current research really predicts that a long-term stress response to predators is actually an adaptive trait in mammals, evolving only under certain life histories, such as intermediate lifespans, cyclic populations where prey and predators undergo intense periods of population growth followed by mortalities, and then it could possibly also be dependent on predator hunting mode. The dusky-footed wood rat itself has a very short lifespan. It only really lives about 1 to 3 years. Its predator is also a constant, predictable environmental stressor. Therefore, having a long-term response to said stressor would not be beneficial to it in any way. It would incur a lot of the negative side effects without long-term benefits. Simply putting it, a long-term response such as that would be maladaptive for a species like the wood rat. There are non-cyclic populations where they experience constant predation risk. Their predator populations remain relatively stable over multiple generations. And this is in contrast to cyclic populations where the prey species will undergo massive booms in population growth, followed by the predators also increasing in size, consuming those prey and causing dramatic declines in their overall populations. This is where we see unpredictable risk come into play. So having such a long-term response wouldn't benefit their offspring. Lastly, predator hunting mode may actually play a major role. Current research shows that prey will respond differently whether they have a dominant ambush predator like a mountain lion or bobcat, which hide and wait for prey to come by, versus a cursorial hunter like a wolf or a coyote that runs down its prey. What we see is that ambush hunters leave more predictable risk cues. They'll defecate and pee in the same spot quite often. They'll hunt in very specific habitat types that are more predictable for prey to assess. Therefore, these more predictable cues are really what allow a prey species such as wood rats to simply respond to a short-term stressor or short-term risk within their environment, rather than being in a constant state of stress. This may change if they were to have a cursorial hunter, such as a wolf or coyote, as their dominant predator. Ultimately, the cost of a long-term stress response on the wood rat would far outweigh any potential short-term benefits. So what really determines whether a long-term stress response exists in a species? Is it the strength of their antipredator defenses, both behaviorally or physiologically, or is it a life history trait thing? Well, my research suggests that life history traits themselves may limit and constrain potential antipredator defenses, especially when it comes to something like a long-term stress response. And that's all I have to say about that.